Hello everyone, welcome back to our channel. Today is the embryology class. Already we have started with the general embryology classes, spermatogenesis, oogenesis, fertilization and the first week of development. And in this video we will see the second and third weeks of development. First we will see the second week development. Second week development of the days 7th to 14th. In the first week implantation started that we have seen in the last class. Now the implanted embryo is more deeply embedded in the endometrium. We have seen in the last class the cells have divided into trophoblast and embryoblast in the first week. So in the second week this trophoblast further develops to form the placenta. Embryoblast gives rise to the bilaminar embryo and two cavities, amniotic cavity and yolk sac. The student should remember, so this is the second week, so the embryo is bilaminar, two cavities are the amniotic cavity and the yolk sac. So in this second week, first we will see the changes in the trophoblast separately and changes in the embryoblast separately. Even though both occur together, for our description we are seeing separately. On the 8th day of development, the trophoblast differentiates into an inner layer, cytotrophoblast, and outer layer, syncytiotrophoblast or syncytium. So this inner layer is having the cell boundaries and the outer layer is not having the cell boundaries as it appears like syncytium. So we have already seen that outer layer is called as syncytiotrophoblast. The trophoblast outer layer is syncytium. So this is the syncytium of the trophoblast without cell boundaries. And the inner layer, this is the cytotrophoblast which has the cell boundaries. So changes in the trophoblast into two layers we have seen till now. So we are seeing the changes in the trophoblast, outer syncytium and inner cytotrophoblast. So the syncytium is growing enormously so that a number of lacunar spaces appear in the syncytium. So this lacuna is communicating around the other through the cards of the cell. So this is the cards of the cells that will be called as the trabeculae. So cards of cells in between these lacunae will be called as trabeculae. You can appreciate the cytotrophoblast is growing in the trabeculae. We can use the other color. So this is the cytotrophoblast growing in between the lacunar spaces as trabeculae. So the trophoblast further, this syncytiotrophoblast further penetrates the endometrium so that breaks the maternal capillaries. So this breaks the maternal capillaries so that the maternal blood reaches the trophoblastic lacunae. You can now appreciate this, all these lacunae are filled with blood. So the syncytiotrophoblast is eroding the maternal capillaries and the maternal blood is reaching this lacunae. Now you can again appreciate this cytotrophoblast are growing in between this lacunae full of blood. So the trabeculae are now called as the primary stem villi. What is this primary stem villi? With a central core of cytotrophoblast and peripheral syncytiotrophoblast. Now this lacunae are called as intervillous spaces. So this is the starting point of the formation of the placenta, formation of primary stem villi. Primary stem villi contains in the center cytotrophoblast and in the peripheral syncytiotrophoblast. We have completed the changes in the trophoblast. Now we will continue with the changes in the embryoblast. Inner cell mass first differentiates into the flattened cells. It is called as hypoblast. According to embryologists, the hypoblast is the first germ layer to be formed. The remaining cells become columnar and it is called as epiblast. Now the embryo is in the form of a two-layered germ disc, hypoblast and epiblast. Simultaneously, a cavity. So this cavity appears between this epiblast and the trophoblast cells lined by the amniogenic cells. So these cells are called as amniogenic cells, and this cavity is now called as the amniotic cavity, filled with Lecker amni, so that the roof of the amniotic cavity is formed by the amniotic gels, and this floor of the cavity is formed by the epiblast. Now we are seeing the changes in the embryoblast. Already two layers have been formed, epiblast and hypoblast, and amniotic cavity has been formed. Then this membrane, this is the Husser's membrane from the hypoblast. Some embryologists suggest whether it is formed from trophoblast or from both. So this Husser's membrane, flattened membrane, lines the inner side of the trophoblastic cavity. So that now there is one cavity formed, primary yolk sac or primitive yolk sac, lined on all sides by the hypoblast. Roof is formed by the hypoblast and floor is formed by the Husser's membrane derived from the hypoblast. Now, in the later part of the second week, a loose network of cells called as primary extraembryonic mesoderm. So, this is the primary extraembryonic mesoderm which is derived from the trophoblast. Why it is called extraembryonic? Because it is lying outside the embryonic disc. So, this is the embryonic disc and this is lying outside the embryonic disc, and it will not give rise to any tissues of the embryo. So this extraembryonic mesoderm appears between the trophoblast externally and the amniotic cavity and the yolk sac internally. And you can appreciate in the extraembryonic mesoderm a number of lacunar spaces appear gradually. So this is how the lacunar spaces in the extraembryonic mesoderm appear. Now they will fuse together to form the extraembryonic coelom or coelomic cavity. So now the coelomic cavity has formed. So this is the coelomic cavity. Splits the extraembryonic mesoderm into two layers, somatopleuric and splachnopleuric. What is somatopleuric? We will take the another color. Somatopleuric is the extraembryonic mesoderm lining the trophoblast, that is cytotrophoblast, and this outside the amniotic cavity. So that will be called as somatopleuric mesoderm. Now we will see the splachnopleuric mesoderm. Splachnopleuric mesoderm is lining the we will take the other color, lining the yolk sac. So this is called splachnopleuric. So this is called splachnopleuric and this is called somatopleuric. Both layers are continuous at the one part, that is called as the connecting stalk, which will form the future umbilical cord. So now we have seen already somatopleuric and splachnopleuric mesoderm has formed, and that is they are continuous at the connecting stalk. Now we should see the important two membrane formation. One is the chorion and another is the amnion. What is chorion? Chorion is formed by this. So these are called as together. Trophoblast and this somatopleuric mesoderm will be called as chorion. So what is this chorion? Will give rise to all these villi which is developing from the chorion will be called as chorionic villi. Another is this amnion. Amnion is formed by this amniogenic cells. So these two membranes again we will see in the further development of the embryo. Now this yolk sac which is reduced by this formation of coelomic cavity will be called as secondary yolk sac. So the yolk sac may be the short notes. We have to write primary yolk sac, secondary yolk sac and tertiary yolk sac subsequently. So now we are seeing the changes in the trophoblast and embryoblast elaborately in the second week. Now it is going to be the end of the second week. One important change occurs, precaudal plate. Precaudal plate may be asked as short notes for the students. So what is precaudal plate? So we have to remember still now the embryo is in a form of a small disc-like structure containing two layers of cells. One is epiblast, that is columnar, and another is flattened cells, hypoblast. It is not known which is the cephalic end and caudal end. This precaudal plate determines the cephalic region of the embryonic disc. What is the changes in this precaudal plate is this hypoblast cells, that is the hypoblast cells near the cephalic end becomes columnar and tightly adherent to the epiblast. So that is called as precaudal plate. So this precaudal plate determines the cephalic end and also divides the embryo into right halves and left halves. So this is what I was describing. This is the precaudal plate. The hypoblast, which is very flattened near the cephalic end, becomes columnar and it is tightly adherent to the epiblast. So this determines the cephalic region of the embryo, and that formation is called as the precaudal plate. Still now, what are the changes we have seen in the trophoblast? Two layers, syncytiotropoblast and cytotrophoblast. Two inner cell mass, embryoblast divides into two layers, epiblast and hypoblast. Two cavities, amniotic cavity and yolk sac. Again, extraembryonic mesoderm form. It divides into two layers, somatopleuric and splanchnopluric, by the formation of coelomic cavity. And near the end of the second week, precaudal plate appeared. So this completes the second week development. Now we will continue with the third week development. Now we will start with the third week development. Important change in the third week development is bilaminar germ disc into trilaminar germ disc. As we have seen in the second week, changes in the embryoblast and changes in the trophoblast separately. In the third week also we will see like that. First we will start with the changes in the embryoblast, and again we will continue with the changes in the trophoblast. Changes in the germ disc is, if you see the shape of the germ disc, in the second week it is circular, and in the third week it is becoming rapidly changes into oval area. That is the major change in the shape of the germ disc in the beginning of the third week. At about 18th day of development, the disc becomes pear-shaped, so that with a broader cephalic end and a narrow caudal end. So we have now the germ disc has differentiated the ends of its own cephalic and caudal end, and the connecting stalk is attached to the caudal end. So this is what we are describing, the pear shaped. So this is the pear shaped, and this is the cephalic broader and narrower caudal end. So now the major change in the third week you can appreciate, this is the primitive streak. So primitive streak appears in the caudal end. Already we have seen precaudal plate appears in the cephalic end. So this primitive streak formation in the third week is an important change, and this will be called as gastrulation. So this primitive streak is very important formation, or it is the primary organizer. So this primary organizer only determines the further development of the embryo, and this formation is called as gastrulation. So it may be asked as short notes, primitive streak or gastrulation. So we are now going to see the formation of primitive streak. Primitive streak is from that is the epiblast or the primary ectoderm. So epiblast or primary ectoderm differentiates into three functional areas. This is as if we are seeing the germ disc from the above. So we are seeing now only the epiblast. Epiblast, if you appreciate, near the caudal end. So this is the pluripotent cells which is now going to form the primitive streak. Otherwise students can understand that the primitive streak is now going to give rise to all the three layers. Otherwise hypoblast will be replaced. Intraembryonic mesoderm will be given by primitive streak. Even the epiblast will be replaced by this primitive streak. So total cells are replaced by primitive streak. So it is the primary organizer of the embryo. So this is the place, these are the cells which are going to form the primitive streak. So this middle part which is going to form the neural cavity or the neurectoderm. So this is neurectoderm, and already we have seen this is the precaudal plate, and these are the remaining epiblast cells. So what is primitive streak? Primitive streak, when does it appear? Appears at about the 15th day of development. What is the shape or structure of the primitive streak? It is a narrow median groove with raised lateral margins. Even though name is a streak, its appearance is as if like groove in the middle and raised margins on the sides. How it is formed? It is formed by the active migration and invagination of the pluripotent ectodermal cells, which we have seen at the caudal end. It is invading into the space between the epiblast and the hypoblast. So that is the formation of primitive streak, and this process is known as the gastrulation. So this is the good picture to show the appearance of the primitive streak as I was describing. It is a groove with raised margins. So this is the primitive streak, and the cephalic end of the primitive streak is slightly enlarged, and it will be called as primitive node or Hansen's node. So if you cut at this level, the embryo will appear as, so this is the epiblast which is giving rise to the primitive streak. If these cells are invading into the space between the epiblast and hypoblast, so these are the migrating cells. So this process is called as the gastrulation. You can appreciate even the hypoblast is replaced and mesoderm is formed. So all the three layers are formed from this primitive streak. So which is now replacing all the existing cell layers in the second week, and this is a very important formation or primary organizer of the embryo, that is called as the primitive streak. So now it is called definitive ectoderm, definitive endoderm, and this is called as the intraembryonic mesoderm. We have already seen extraembryonic mesoderm. Now this is very important layer, intraembryonic. It will give rise to the embryonic tissues. This picture also clearly shows this primitive streak, primitive node, and this is that two-dimensional picture which is given in all the embryology textbooks. You can appreciate. So this is that gastrulation process. This gastrulation replaces all the three germ layers, giving rise to the definitive germ layers. So this primitive streak, that cephalic end is called as Hansen's node, and we have already seen this is the primary organizer. What this primordial germ cells, that is primordial germ cells, we have seen in the spermatogenesis and oogenesis classes. From which part of the primitive streak? From the caudal end of the primitive streak it appears. So this primitive streak starts on the 15th day, and it is the primary organizer, and it also gives rise to the primordial germ cells. So we are seeing the changes in the embryoblast or the germ layers in the third week. We have already completed the important primary organizer formation, that is the primitive streak. Now the primitive streak gives rise to the secondary organizer, notochord. Notochord is again the important short notes for the students. How it appears, when does it appear? It appears on the 17th or 18th day of the development. How it starts? We have seen that primitive streak is containing the primitive node. This primitive node presents a depression that is called as primitive pit. So this primitive pit, if you see, this is the ectoderm and this is the endoderm. We will take another color. So here is the primitive streak and this is the primitive pit. So this primitive pit grows like this. So that process is first called as notochordal process or head process. So it is containing that central canal. So that is called as notochordal process or head process, that is the beginning of the formation of the notochord. So this is what I was still explaining. This is the primitive streak and this is the primitive node. Primitive node presents a central depression called primitive pit, so that this primitive node grows in between these two layers as the head process. Head process or the notochordal process, which is containing the central canal. Now we are seeing the formation of the secondary organizer or notochord. So this is the primitive streak and this is the primitive node. In that there is a central process. Primitive pit enlarges as notochordal head process with the central canal. So this is called as notochordal process or head process with the central canal. And this is above lined by amniotic cavity and below lined by the yolk sac. At one stage, we will take another color. So this adjoining the yolk sac, this disappears so that communication occurs, and that is called as the neurenteric canal. So it is appearing like one plate. So this will be now called as the notochordal plate. And then that will be replaced by the solid cord of cells, that is the notochord between the amniotic cavity and the yolk sac, that will be called as the definitive notochord. So these are the stages in the formation of the notochord. So this is what I was describing already. So this is head process. This is the head process containing the central canal. At one stage these cells disappear. So this canal is now communicating the amniotic cavity and the yolk sac. That canal will be called as the neurenteric canal. So that now that plate will be called as notochordal plate. This three picture depicts the development of the notochord. As I was describing from the beginning, primitive pit. So this primitive pit now grows head process with a central canal. At one stage, both cavities are communicating. That is the neurenteric canal. At this stage, the notochord appears as a plate-like structure called notochordal plate. And lastly, the definitive notochord appears between the definitive ectoderm and the definitive endoderm. And also the intraembryonic mesoderm has already formed. This picture is a good picture of gastrulation. The primitive streak replacing the hypoblast, forming the definitive endoderm, forming the intraembryonic mesoderm, and also displacing the epiblast into definitive ectoderm. So the primitive streak only gives rise to all the definitive three germ layers. So we are completing the changes in the germ layers in the third week. We have seen two important organizers formation, primitive streak and the notochord in the third week, which are very vital for the development of the embryo. Third week development, primitive streak and the notochord. Without these two organizers, the embryo will not develop properly. Now we will continue with the changes in the trophoblast. The changes in the trophoblast in the second week we have seen, that is the primary villi formation with a central core of cytotrophoblast and surrounded by the syncytiotrophoblast. Now in the third week, that is the formation of secondary villi and the tertiary villi. So this is what we are seeing. So in the beginning it was primary villi containing the central core of cytotrophoblast and the periphery of syncytiotrophoblast. Now you can appreciate extraembryonic mesoderm grows into this primary villi. So what are the layers now? Central core of extraembryonic mesoderm surrounded by cytotrophoblast and syncytiotrophoblast. Now it will be called as the secondary villi. So this is the good picture, enlarged view of the formation of secondary stem villus. So secondary, this is maternal side and this is fetal side. So maternal side we are seeing this is the intervillous spaces filled with maternal blood. So now the villi are growing, that is from chorion. What is chorion? Extraembryonic mesoderm and trophoblast will be called as chorion. So they will be giving rise to the villi. So now this is the extra embryonic mesoderm growing into the central core of cytotrophoblast and now surrounded by the syncytiotrophoblast. That will be called as the secondary stem villus. So students should understand what is it all formation, what is the purpose they have to think. So here the maternal blood is lying in the lacunar spaces. So the fetal blood vessels should reach the stem villus so that it can take this maternal blood. So that is the formation of primary, secondary, tertiary, all these villi formation. In the third week, after the secondary stem villus, tertiary stem villus is formed. How it is formed? So in the extraembryonic mesoderm, so these are the fetal blood vessels in the extraembryonic mesoderm grows in the core. So students should understand now, it is very clear, this is the maternal blood and this is the fetal blood. So that is the formation, that is the function of the formation of the all these stem villi. So in the tertiary stem villi, in the core is the fetal blood vessels surrounded by extraembryonic mesoderm, surrounded by trophoblast, cytotrophoblast and syncytiotrophoblast. In the third week, we have seen what are the formation in the germ disc is 3 germ layers and the primary organizer primitive streak is formed. Then the primitive streak has given rise to the important secondary organizer notochord. This third week development will be called as gastrulation, very important change. In the trophoblast side, we have seen secondary villi formation and tertiary villi formation. I have covered very minute details of development of second week development and third week development of the embryo. I hope you should have understood. Any doubts you can post in the comments. Those who have not subscribed to the channel, do subscribe now, as our channel contains gross anatomy, histology, embryology, neuroanatomy, quiz, practical section, radiology section, so many playlists in our channel. Do subscribe now. Keep watching the videos. Thank you.